intractable diseases which cannot be cured by medical methods. Fourthly, everyone ought to know the rejuvenation of tissue organs. Everybody should not be superstitious in the regenerative substance announcement today. We did not create any new ingredients, new drugs, or new chemical compositions, but we only reprocessed our daily meals into something that can activate our regenerative potential. It would be unnecessary to explain, and we could easily get regenerative restoration if food from nature can activate regenerative potential. That is why we need to extract it from our daily meals. How does one extract it? We extract it by depending on cell culture to transform into tissue organs, which was announced in 2002. Until now, there are few in the world can achieve it. We have begun a series of experiments to get a regenerative nutrition spectrum. In other words, we have known the secrets of what cells should be fed and transform the secrets into a spectrum. Then, according to the spectrum, we calculate and code each ingredient to finally form regenerative substances. The regenerative substance is something people can take daily and even is a must for people to have daily meals in the future. We use 3 models to accomplish the process. Since most of us here are not the professionals, I will explain the process briefly. First is the model of somatic cells. We use somatic cells, not the stem cells. Somatic cells are derived from the intestinal epithelium, which are cultured into tissue cells and transformed into stem cells and then into tissues and organs. Nobody else in the world has achieved such a result. Some has just finished part of it in picture 6. In 2002, I announced the achieved results, but everyone said that it was impossible. However, all of the studies and research around the world now are following our routes. The scientist who cloned Dolly, the sheep, also declared that he began to focus on transforming somatic cells into stem cells, instead of reproducing cloning. All of them have few results, but we have achieved it. Through this, we get a nutritional spectrum which is like a prescription. It is also like cooking, where we know what to cook with various raw materials. The other model is tissue organ regeneration from tissue explants. Nobody else has achieved it. We directly culture organs and detect the nutrients required during tissue organ regeneration and establish a nutritional spectrum. This is the second kind of spectrum. The third spectrum is obtained directly from the human body. We take some human tissues in hospital operating rooms and culture the tissue organs in vitro. So we obtain 3 kinds of regenerative nutrition spectra by combining the results of animal experiments and human body experiments. These 3 spectra are also the foundation of regenerative substances for the human body. Making regenerative substances based on these spectra is a big project of nutrology and bromatology. In the future, 1 man's serving of food support 3 people. The diet became refined, which means we analyze the composition of regenerative nutrients and code it, and use these codes based on the spectra. Then what is the code? Each composition has a spectrum, and list every composition spectrum. Then we will see what the cells need during its life cycle, and in the end a coding is formed through computer programming. Take amino acids, for example. There are several amino acids in our human body. Besides non-essential amino acids, we have to obtain some amino acids that cannot be synthesized by ourselves through intaking foods. Thus, we encode all kinds of amino acids and then the cell's needs, such as which is the first to be taken and which is the last to be taken, what the cells need when you are excited and what the cells need when you are sleeping.